Question 5, model answer. Really tough, this question, really, particularly if you're not fascinated with plants. Some bits of it you should get, though. Some bits of it you should get. Name the parts A to D. So we're in A01 territory. So just naming them. So A is the outside part, the seed coat or the tester. Right, there's B and C and D. Let's do D first. D is the living part. It's the baby plant, as it were. So we've had a egg and pollen fertilized zygote. So now this is the growing part, the baby plant. So this is the embryo. Or we could say, the first bit of growth that we're going to get, we could say radical. But embryo is the best answer there. And now we've got B and C. So the clue for C could be, well, there's one of them there in the maize, which is like a grass, whereas the other two, broad bean and castor oil, they're kind of broad-leaved plants, and they've got two of these. So mono and di might give you a little bit of help there. So these are cotyledons, or the seed leaves. So mono cotyledon, di cotyledons. So I can't even spell it. Cotyledons. And B is just a store of starch, and that's our endosperm. So you needed all of those to get 2. 3 of them right gets you 1. So if you got 2 right, my apologies, you don't get anything. And now, similarity with the castor oil seeds. You really need to know these names to get down here. This is AO2, but just getting information from the diagram, really. So broad bean and castor oil. Let's avoid the things that are true for all 3 of them, because that's not really what they're wanting. So what's just true of the broad bean and castor oil? Well, we could say it's got 2 of these cotyledons. They're both dicotyledons. So have 2 cotyledons, or they're dicotyledonous, which is a great word. Difference? Well, we don't have much of this white area, which we identified as endosperm. So the castor oil has endosperm, the broad bean has no endosperm. Broad bean has no or little endosperm. I'm going to talk about it from the broad bean point of view, because that's what it's asking. How is the broad bean different from the castor oil? How is the maize similar to the castor oil? So this one and this one. Well, they both have a big endosperm. Both have endosperm. The difference between the maize and the castor oil, well, 1 cotyledon, 2 cotyledons. So the maize is a monocot. So maize is a monocotyledon. I'll be tempted to put in, even though we're talking about the maize here, I'd say maize in the answer just to make it absolutely clear. And you've got 1 mark per correct row. So if you got a diagonally right answers, again, nothing. Part B. This is an experiment essentially a bit like your methylene blue experiment, where they're testing for dehydrogenase activity. And so where you're getting respiration going on, using the food, digesting the food, respiration, then you're going to get the colour change, and it says it will stain red. So where is it going to stain red shade? Well, it's going to be in the living area of the plant that we're going to get respiration going on. So around the edges of this embryo. So anywhere really, if you shaded the embryo, then you got the mark. But particularly around the tips of this embryo. Then we've got an experiment that you have done or you have seen demonstrated in the lab. I saw these out, and it's in your lab book. So you do need these lab book experiments, they do crop up. Name the enzyme responsible for the clear zone around the unboiled seed. So this is starch, and the starch disappears. So it's the enzyme that breaks down starch, and there's only one enzyme that does that, and that is amylase. And that's what's happening over here. So we're getting this starch broken down into maltose and then glucose, and then can be used in respiration. What was the purpose of the boiled seed? Well, boiling it will denature the enzymes, will denature this amylase. It's to show that this effect is due to these kind of enzymes in living seed, rather than just having the seed sat on there or some other effect. And that's what we call a controlled experiment. Controlled experiment, not a controlled experiment. You might have controlled variables. So control experiment is proving that the enzymes cause the clear zone. Proving enzymes cause the clear zone. Part D, how the two enzyme systems depend on each other. Now this is really tough. So this is one of those questions aimed at A-star candidates, hoping that give them something to munch on. So we've got two enzymes. We have got the amylase, and we've got the dehydrogenase. So you really need to identify those. And the amylase, that's breaking down the starch, and that provides a substrate for the dehydrogenase to work on. I know it's a really kind of horrible thing to say that, but amylase goes before the dehydrogenase, because the dehydrogenase is removing hydrogens, and the hydrogens are currently stuck on this carbohydrate, which is being broken down by amylase. So provides a substrate for dehydrogenase. You might argue that, well, it's going to get turned into the starch has got to go into maltose first and then into glucose before any dehydrogenase takes place. And then why does amylase depend on dehydrogenase? Now this is very tenuous. Well, these dehydrogenase, these hydrogens are gonna disappear off to generate ATP. In the previous question when we talked about that, using hydrogen carriers. So that's going to give us some ATP, and what's ATP used for? Growing the plants and so on, but to make amylase as well, to synthesize cell components. So dehydrogenase allows respiration to provide energy for the synthesis of amylase. I'm not quite sure I might have got that without looking at the mark scheme. A tough question. I'm Max.